next we're going to talk about, I mentioned the CSF, the cerebrospinal fluid already, and its role of supporting and nourishing and protecting, cushioning the central nervous system. We're going to take a look at a schematic, a highly schematized view of the skull and the brain and the CSF. And I'm going to start this discussion by pointing out something remarkable. You learned early on in this course that the body is hollow. It's a striking feature of the body that is kind of unexpected, I think, for students of anatomy, students new to anatomy, when you find out that the body has all these hollow spaces inside that are then filled with viscera or whatever. The brain itself, it turns out, is hollow and has inside it fluid-filled cavities that the brain is kind of wrapped around. And so the same principle applies here. These hollow spaces inside the brain are called ventricles. And so we're going to now talk about the cerebrospinal fluid and the cerebral ventricles, the CSF-filled ventricles. So here's the layout of our schematic. We have the skull, the cranium, actually, in gray here. Notice our dura in green, right up against the skull. Just underneath that, we have the arachnoid inflated with CSF. So this space in here is going to be CSF-filled. It's the subarachnoid space, and it's got lots of CSF in it. Where does the CSF come from? Well, it's going to originate in the ventricles. And so this schematic just has one cavity there that's simulating, that's approximating all the ventricles. There's four ventricles, four cerebral ventricles. I've just lumped them into one structure here. We'll look at the anatomic detail in a little bit. So how do we make CSF? It works like this. We have blood, pressurized arterial blood coming into the brain. And so we have several arteries that come into the skull, like this, into the cranium. And they will supply blood, of course, to the brain itself, the neural tissue. But some of the blood will go into capillaries that are located inside the ventricles. And these capillaries are special. They're a lot like the glomeruli at the kidneys, actually. You get a similar effect. These capillaries are enshrouded in the structure called the choroid plexus. And what it is it's sort of like, imagine this bed of capillaries here, which is drawn way out of scale. Imagine the pressurized arterial blood arriving in these capillaries. And the way this choroid plexus wraps around the capillaries, it makes them like a little sprinkler. So red blood cells can't get out. They stay in the bloodstream, and maybe large proteins are restricted. But blood plasma just kind of squirts out through tiny little gaps in the choroid plexus. So blood plasma can leave the capillaries and enter the ventricular space here in the form of CSF. So the choroid plexus is a filter that allows blood plasma to squeeze out of the capillaries and make CSF. Now, having generated our CSF from filtered plasma, the CSF is going to circulate within the ventricular system, and we'll go over that in anatomic detail in a little bit. At some point, it's going to find a little hole that we call an aperture, not a bold term, but that's what it's called. It's going to find one of three little holes called apertures, where the CSF can actually exit the brain proper. It can exit the ventricular system and work its way outside the brain, still inside the skull here, outside the brain and into the subarachnoid space, where it then is kind of circulating around the brain. Some of it drops down and goes around the spinal cord as well. Circulates around the brain, and eventually, after washing over the brain and supporting it, cushioning it and all that, it can find its way to a little one-way valve here. This is called an arachnoid granulation. And the arachnoid granulation is a little one-way valve that allows CSF to, having come from the bloodstream here, it allows CSF to return to the bloodstream via a cerebral vein or a dural venous sinus. And notice here that I've colored the dura in. Notice here, here's the dura. It's actually, this little dural venous sinus is actually sandwiched between two leaflets of dura here. So it's completely surrounded by dura. And we have our little one-way valve where the arachnoid kind of pokes through the dura and allows pressurized CSF. As long as the CSF is under higher pressure than the venous blood, it can go through here. If for some reason the venous blood gets pressurized and the pressure in the venous blood is higher than CSF, then this valve will snap shut, and we don't let red blood cells go into the CSF, and you can't go the other way. It's one way. So the CSF normally will filter back it will normally be returned to the venous circulation here through these arachnoid granulations, which are like little one-way gates into a dural venous sinus. Venous blood then drains from the dural venous sinuses and from the cerebral veins proper, draining the brain directly, and out of the skull like that. So that's the mechanism by which we make CSF, and we make it, I think it's something like a rate of 2 liters per day or something like that. So, you know, over the course of the day, we make a generous amount of it. And of course, it has to be generated here and then drained here at the same rate to keep it constant, stable level. Okay, now that was a schematic. Let's look with some anatomic reality at the ventricular system. This is kind of an odd-looking image here from Complete Anatomy. The screens that are set up in Complete Anatomy, of course, gives you the benefit of going in there and spinning things around. What I've done here is I've stripped away the skull. I've taken away the entire left cerebral hemisphere. You can see the right cerebral hemisphere kind of ghosted over here. There is a big dural fold right here, a big midline dural fold. There's another one sticking out to the side here. So ignoring all that, what you see is a large horn-shaped lateral ventricle. It's shaped like a ram's horn, or like a C shape like that. And that's one of 2 lateral ventricles. That's our left lateral ventricle. There's one on the right as well on the other side of midline, and it's kind of, you can see it kind of tucked in there. So there are 2 lateral ventricles occupying kind of the deep portion of the cerebral hemisphere. And then from the lateral ventricle, choroid plexus in the lateral ventricle generates some CSF. It can leave the lateral ventricle trickling down through a small passage called the interventricular foramen. Foramen means hole, interventricular because it's going between ventricles. And from the lateral ventricle, this one or that one, we drain down through that passage into a midline singular third ventricle. Okay, so the lateral ventricles are 1 and 2, but we call them lateral ventricles. The third ventricle is here, and it also has choroid plexus. You can make CSF right there in the third ventricle, in addition to making it in the lateral ventricles. And then from the third ventricle, CSF tends to trickle downward like this through another narrow passage called the cerebral aqueduct. And from the cerebral aqueduct, we then drain down and enter the fourth ventricle. And so this one also has choroid plexus. You can make CSF right here. And then I haven't labeled them, but see that little, there's like a little shark fin coming to a point there. There's another little point down there. That's where the apertures are. So CSF that's generated all the way up here in the lateral ventricle comes down through the interventricular foramen, third ventricle, cerebral aqueduct, fourth ventricle. This is where it can sneak out of the ventricular system and outside, and get outside the brain into the subarachnoid space. Okay. So that's the real anatomy of the ventricles. Coming back to our schematic of the ventricles, what's the point that I was going to make here? So I think something bad's going to happen. Right. Okay, so see this aperture? You can imagine a constriction or compression of that aperture right there, or within the ventricular system itself, if we were to constrict or compress one of the passages that connects one ventricle to another. So if we constrict if we occlude an interventricular foramen, or if we occlude the cerebral aqueduct, CSF is going to be trapped and unable to drain to the next point in its destination. And recall that we've got pressurized blood sprinkling out plasma to make CSF, and this is going on at the choroid plexus in all four ventricles all the time. And that CSF has to get all the way back up here in order to leave and return to the venous circulation. So if you block the passage, if you block these apertures, or if you block one of those passages between the ventricles, the choroid plexus upstream of that occlusion is still going to make CSF, and that CSF is going to accumulate. And you're going to get the accumulation of CSF is going to it's going to cause hydrocephalus, is a condition where the accumulation of CSF pools and the ventricular space enlarges and compresses neural tissue to either side, and it's super dangerous. You have to once you realize this is happening, you have to either deal with the acute occlusion or provide a shunt, a passage, surgically implanted passage for CSF to drain out via a separate pathway to prevent increasing accumulation of CSF in a trapped space and increasing damage to, more severe damage to neural tissue.